All right, so we're gonna dissect some wing discs from Drosophila larvae. So this would be if you either want to do in situ hybridizations, or if you want to do an antibody stain, or collect larvae to look at GFP expression or other fluorescent proteins and things like that. So this is in third instar larvae, but like you can do this on younger larvae as well. And if you go a bit later to when they've started to pupariate, then it'll be a little bit tricky because the pupal case will be a little bit harder to get into, and it causes a lot of pressure to build up inside the body, and that essentially you're more likely to cause your guts to spew out everywhere, and in particular like brain tissue and things like that to come out. So you got to be a little more careful if you're into the pupal stages. But so for today we're just looking at third instar larvae. So we've got basic set of equipment that's out here. So you've got our bottles in this case that have some larvae that are crawling up the side. If you wanted to be strict on timing, a good way is to find larvae that are in the white prepupa stage, where they've settled up on the side of the vial and started to invert their anterior spiracles, but are not yet crunchy, that they're still a little bit squishy, that you can squish them with the forceps. And for our purposes, just for demonstration, I'm just going to pick ones that have mostly settled on the side of the vial, not necessarily being too strict about white prepupae for this demonstration. So we've got our flies, and then we've got some 1x PBS, some 1x PBT. The PBS is basically what we'll be dissecting the larvae in, and then the PBT is for washing off the fixative afterwards. In this case, the PBT is good if you're going to be doing an antibody stain or going to be looking at GFP expression. If I'm going to be doing an in situ hybridization on this tissue, then typically before the pre-hybridization steps, I typically will get them out of aqueous solution and move them into methanol for washing off the fixative, and then into ethanol to store at -20 before it actually comes to then doing the in situ process. So in this case, since we're going to go through just if you're going to do an antibody stain or GFP, then we've got PBT, which is 1x PBS plus 0.1% Triton as our wash. And then have Eppendorf tubes that have our fixative in there, which is 1x PBT, again with the Triton at 0.1% concentration. You can go a bit higher to 0.3% that can work good for some antibody preparations, and then it has 4% paraformaldehyde in it. We've got our dissection tools in terms of our scissors, and then some nice forceps that have pretty decent points on the ends, and try and find ones that are have even ends that you can go and pinch something, as opposed to those that may have broken ones on there that might need repaired. You don't want to use those in this particular circumstances. And then a clear glass viewing dish, and then of course our timer for the fixative and wash steps. And so to get started, what I typically do is put PBT in all of the wells. And then what I like to do is have the wells on the outside edges be where I will pick the larvae off and put them into these outside wells and do the dissection within the outside wells. And then for each larvae that I finish, then I'll pick it up and move it into the clean center well. And then once I've gotten all the larvae, then I'll pull them all out and then put them into our tube with fixative. So let's first go ahead and get some larvae, and I will typically do anywhere from 10 to 30 larvae at once. It's really up to you in terms of the number that you can do in a relatively short period of time that you can dissect in a good like 30 minutes or so. looks like I got some flies that are coming out of here, so I'm going to dump these out real quick to make sure I don't have flies coming out all over the place. Okay. Alright, so I'll pick out some larvae. Okay, so we've got our larvae in the dish. Let's stick that under the microscope. And then have all of my tools there in front of me. And I'll go ahead and look at the larvae, get them in focus. Okay, and so then the basic strategy is that you have a larvae and it has its little mouth hooks on the end. And so what I'll typically do is then take one pair of forceps and then grab the larvae by the mouth hooks. And so then as I'm holding the larvae, then we'll take the scissors and cut them about halfway down their body length, and then that'll cause their viscera to stick out of the place where it was cut and essentially pull those away. And largely it's a lot of fat and intestines we pull out with the forceps. And then after that, with grabbing the head with the forceps, then we'll take the other pair of forceps and then essentially kind of flip the larvae inside out like a sock. And then that will expose all the imaginal discs to put them on the outside, so they're exposed to the fixative, so that then it would be available then for looking at. So then what we're not going to be doing today, we'll put it in another session of what to do after the fixation process to pull the discs off of the carcasses, to then be able to put on slides to look at. So for now we're just doing the dissection process. So I've got one larvae that I've grabbed, and sometimes it helps to use one pair of forceps to just kind of grab about a third of the way down the body, and then use that to then position the other pair of forceps so you can effectively grab the mouth parts, and then go and take my dissection scissors and then cut it in half. And then now use the other pair of forceps while I'm holding the mouth parts to take and pull out the viscera. The fat is kind of white and fluffy, and then you can see more clear stringy parts, which are the intestines, and then also some of the tracheal bits. And so I can go ahead and take these forceps and then turn it inside out. And then pull off the guts, or pull off some remaining fat off there so that the carcass is kind of cleaned up. Alright, so then I've got one, and then I'll go ahead and then transfer this guy up over there to our middle section, and then we can grab another one and do the same thing again. Grab it by the mouth parts, use the scissors, cut it in half. Pull out the fat and guts that you see sticking out. And then turn it inside out, and then transfer it up into the upper one. Okay. So that was seeing the process from up there, and what I'm going to do is attach the camera onto the oculars so that you can actually see what we're doing. Alright, so here we've got our third instar larvae that are down inside our dish in PBS, and I've dissected a few of them already, so we can kind of clear this out of the way. So there's like some fat that's down in here, and what we can point out is, say, this larvae right here. This is one that is pretty close to becoming a white prepupa, so it's extended out its anterior spiracles and is not moving as much of the other ones. Some of the other ones are a little bit younger. So this is a nice prime age. This should have some nice big imaginal discs in order to pull out. And then, whereas this one is not quite as old. I think we've got another one that's up here. That one's a little wriggly, doesn't have its anterior spiracles extended. So let's focus on this one here. And so we can go through the process. So we've got 2 forceps here, and we'll move the other larvae out of the way. And then what we're going to do is just kind of hold it in place with one, and then I can use the other pair of forceps to grab the mouth parts like that. Okay, then I'm going to take the scissors and then sometimes if they're wiggly, I kind of use the scissors to straighten out their body. But in this case, since this one's a little bit older, it's not wriggling too much, but then basically then come about halfway down and cut it. And so now I can see that we've got some white parts, that's the fat, and then more clear parts, those are some of the intestines and other parts, and I can basically just grab these and pull them off. It's actually a little bit trickier to do it while I'm watching it through the camera, because I'm very used to just doing it on the microscope through the oculars. So it's a little bit different. Okay, got it. That's pretty clean. Okay, so then now comes the trick. So this is often the part that just requires a little bit of practice. So then essentially what I'm going to do is then I usually start kind of a little bit closer to where the other pair of forceps are, and then just kind of gently grab it and then slide the cuticle up and over the other pair of forceps, and then now we've got a larvae that is inside out. And so then I can use the other pair of forceps to slide it off. And so we've got some fat sticking off a little bit up here off of some of the salivary glands, for example. So I can hold it in place and trim this off as best I can. Sometimes you don't get all of it off, it's okay, but it's best to clean it up as best you can, because sometimes the fat can absorb some of your stain, and it's best to try and clean it up as best as possible. But you want to try and work as efficiently as you can. Okay, so then I'll go and take this guy and pull it out, and then put it into the bin up above. And so we can go and see the bin up above. And so here are our larvae that we've dissected. I'll go ahead and do one more. So here's another one that has the anterior spiracles out, so that one should probably not move around as much. Move the other larvae out of the way. So just kind of gently grab it, and then identify the mouth parts. Take the scissors and snip it about in half. To remove as much of the fat as possible. That one's a little bit cleaner. And then go ahead and flip it inside out. And then here's another one where I can try and pull this fat off here. I try not to mess with the trachea, which is what the fat is attached to, because the wing disc, leg disc, and the haltere discs are often connected to the trachea. So if you lose the trachea, sometimes you can lose those discs. Here's another little bit of fat. And try and get off. Alright, and then we can look at the other side. Yeah, so overall that's pretty good. So I'll just pull this one off and bring it up again. Again, let's have a go look at those and then just see what they look like. Maybe zoom in a bit and we can see if we can point out some of the discs. Okay, so we mentioned that the trachea here is often has the wing disc attached to it, and right there is the wing disc. So that you have one wing disc, and very likely you have the haltere and leg disc attached there. Up here is the brain, so you have the two lobes, first lobe, second lobe, of the optic lobes, and then that's attached to the ventral nerve cord that's attached as part of it. And then have a set of eye discs and leg discs that are up there. And then if we flip it over to the other side, maybe I need my other pair of forceps to do so. Flip this one over. So you can also see that we've got a bunch of nerves that are coming off of the brain up above there. And on the other side, we have the other wing disc. So yeah, so we've got this wing disc over here, and then there's another wing disc and haltere disc up here. So they kind of stick out to the side like the larvae is wanting to hug you. And then up above on this part here, we've got where the brain is. And so those are where the main discs are that we're interested in staining for a lot of our purposes. Okay, so then now that I've got a set of larvae that I want to look at, then I'll go ahead and pick them up and grab them with the forceps. Try to usually not grab them right on the discs, and then I'll take them and drop them into our tube that has the fixative. And then go ahead and set our timer. And this is set for 30 minutes. That's usually a good amount of time for fixing. And then in the meantime, while those are fixing, then I can dissect some more larvae. Alright, so our fix is up, And so you notice that the larvae are all down at the bottom, and so then what I then do is take a pipette so another glass pipette for waste, and then a waste container. And then I will just remove most of the liquid above the larvae and put the fixative in the waste container, and then squirt in our PBT for our wash. Trying to be pretty gentle, but the larvae do come up as you put the liquid in. And then usually when I remove the fix, I will typically do one quick rinse, remove the liquid one more time, and then add another rinse, and then we'll let it wash the fixative off for about 10 minutes, and then do 5 washes for 10 minutes. And then once you've gone through the PBT washes, then you're going to be ready to add your block if you're going to go through a block, or you can then immediately add a solution containing glycerol and or some anti-fade stuff if all you need to do is look at the GFP signal, and then you can pull off the wing discs and start to put them on slides. Let's take another one. Let's dissect that one there. This one's being a little bit squirrely. Got it. All right. let's imagine that we'd finished the fix and wanted to pull off the discs because we wanted to mount them on slides. So again, down here at the bottom, that's where the brain is, and up here we can see the trachea, and there's actually, we can see the wing disc pretty clearly up here. And so then if I wanted to pull the wing discs off to look at them under the microscope, I got the other wing disc. I can grab down, the other wing disc is over here, this guy there. And so typically you can just kind of tease away the disc. I don't like to grab them very much because they are very delicate, but you can just kind of gently tease them off, and then if you do that, we've pulled off a wing disc. And zoom in, or yeah, there is our wing disc. And so then if I wanted to then put that on a slide, just very carefully grab it with the forceps. And then go and pick it up, put it on our slide. So the other thing that we can note so here's an example of a brown pupa that is much too old typically for dissecting. It's floating up because it's got some air trapped inside there. Let's see if I can grab it and pull it down. So now I think you can already see that in just trying to grab it down, that as I've accidentally grabbed onto it a little bit too hard, I just squeezed on it, I guess, a little bit too hard, and already broke the pupa case, the cuticle lining around here by the operculum, and then now we have all this like fat and stuff that's coming out there. So that demonstrates what why the larvae are a little bit better than trying to go after pupae. All right, I think that's good.